All right, it's 5 o'clock. We will start with the webinar today. First of all, warm welcome to all participants and to, of course, also to our panelists today, which I will introduce in a second. Also, welcome to Georgia, who's co-hosting with me today from the CUT Europe in Constance. And it's my pleasure to introduce two absolute experts in the field who basically spent their entire careers on microfluidics and tissue engineering and also sensors and of course organ on chip. And we are really thrilled to have a topic for today's webinar, which is really easy access. So everybody, even if you're never worked with organ on chip before, you will be able to follow. And I think it's important that we also talk about how to actually get started, because there is so much cool organ on chip science out there and it all sounds so fancy, but sometimes it's really a bit of a burden that you're like, oh God, I don't even know where to start. So we try to tackle this issue a little bit today. And it's my great pleasure to introduce the two panelists today. So first of all, we have Julia Rogal. Julia is, as I mentioned, a microfluidic and tissue engineering expert who really spent her entire career ever since her postdoc in Tübingen at the university on this topic. She then afterwards moved to Sweden, actually to the KTH Royal Institute of Technology in Sweden in 2022, and she stayed there until summer 2023. And by that time, she actually was awarded with a Marie Curie postdoc fellowship at the Karolinska Institute in Sweden, where she, up to now, develops iPSC-based organ on chip models for neural research. And then we also have Thomas Winkler. Thomas is also a bioengineer and, like I said, also an expert in especially microfluidics and sensors. He shared with Julia quite some time in Sweden, where he was a postdoc also at the KTH Royal Institute of Technology, also with a Marie Curie fellowship, where he started adding cell culture to the mix. So he also knows about this topic. So feel free to save all your questions that you have for both of them. And then in 2021, he actually established his own research group back in Germany at the TU Braunschweig. And really exciting, and congratulations at this point, Thomas. He was just awarded with an ERC starting grant to work better on controlling the chemical environment inside organ on chip systems. So I think I promised you enough, and I'm really excited to get the webinar started.
All right. Hi, everyone. I'm Julia. I'm very excited to be here today. I'm also very happy to share this webinar with my colleague Thomas. And we really hope that we get some of you started in the organ on chip technology today. And I think before we'll start with the slides, we'll have our first poll. All right, so first question would be, what is your background? And this is a multiple choice question, so you can select several answers. And then also our second question would be, what do you actually picture when you hear the term organ on chip? Looks like we have a lot of PhD students in the area of life science and natural sciences, some master students, some bachelor students, also many others, and few people from social science and from engineering. All right, should we then get started? Let's maybe start with the question of why is an organ on chip actually called a chip? Because obviously there's quite significant differences to computer microchips, right? But historically, the first organ on chip were fabricated with methods that are actually originated from photolithographic etching, which is a method that is also used to manufacture computer microchips. And this is why organ on chip is called a chip. And why do we need organ on chips? Because there's really a need for novel models to study human physiology. For example, in the drug development pipeline, which is extremely long, extremely inefficient, and extremely expensive. And especially a transition from preclinics to the clinic is problematic, which is really highlighting that conventional preclinical models are not relevant for human physiology. So what are conventional preclinical models? On the one hand, we have animal models, which do have a complex tissue architecture, and you can model systemic effects. However, it is often impossible to translate findings from your animal models to human physiology. On the other hand, we have conventional 2D cell assays, which do have that human genetic background, and it's easy to standardize them, but they do lack complexity. And organ-on-chip, as part of a group of model systems that's called microphysiological systems, actually combine the advantages of both animal models and 2D cell assays. So microphysiological systems are human-engineered tissues in a microphysiological environment. And these systems really aim to recapitulate the in vivo structure and function of certain organs. In other words, microphysiological systems can also be seen as bioengineering breakthroughs that reproduce organ architecture and function in vitro. And there's a broad variety of especially human models and test systems out there. And organ-on-chip is only one of them. And now to complete that picture of non-animal model options that are out there, on the one end, we also have the conventional 2D cell assays, which can also be human, of course, and then we have in silico models, but both of them don't really count as microphysiological systems. And today, in this webinar, we'll of course focus on the organ-on-chips. Now, as Silke promised in the beginning, how would you go about developing an organ-on-chip system? And for that, the human body serves on the one hand as a structural blueprint, and on the other hand as a cell source. If you then combine blueprint and cell source in a specifically tailored microfluidic platform, you'll get your organ-on-chip system. And here are some examples of organs-on-chip. So the development process of an organ-on-chip is a really interdisciplinary effort. And what we're going to do today is provide you a little bit of guidelines if you are thinking about developing your own organ-on-chip system. And the development process starts with your idea, or in other words, a defined scientific question that you want to address with your organ-on-chip model. And for that, we have seen that the human body serves as a blueprint and as a cell source. Then you would move on and come up with a specific concept and a design of your organ-on-chip model. After that, the development process divides up into two branches. The engineering branch is about fabricating your microfluidic device, selecting suitable materials, and you'll also have to tackle the question of whether you want to integrate any sensors or actuators into your microfluidic platform. On the other hand, we have the biology branch, which is about identifying suitable cell sources for your organ-on-chip model and also about preparing those cells. And you'll also have to address the question whether you need to integrate any biomaterials or scaffolds in order to really realistically replicate the in vivo situation in the human body. These two branches later on merge again when you inject your cells or tissues into your microfluidic device. And finally, you need to see what's happening to your tissues in the chip. So you need to develop some readout methods that allow you to see what's happening. And in the last step, you need to functionally validate your model, meaning that you have to show that the model that you developed in this entire process is actually capable of addressing that defined scientific question from the beginning. And then your model is ready to be applied. Now, while this whole process looks very straightforward in this graphic, let me assure you it is not. So in each step of the way, you have to be prepared to go back one step or even several steps. And now let's walk through the process again, but with a little bit more detail. So regarding your defined scientific question, in this step, you really need to define an intended application purpose of the model. In other words, a context of use. If you just take away one thing from that webinar, please let it be that if you develop an organ-on-chip model, that the model has to have a specific context of use that you better define right before starting to develop that model. And in this step, you also define how do you actually recapitulate the minimal functional unit of your target organ in the microfluidic device. Next, you will come up with your specific concept and the design of your chip. And in this step, the key challenge really is that you design a platform and a procedure that is as simple as possible, but as close as feasible to the respective in vivo situation. And to do so, you have to have a very, very thorough knowledge of, on the one hand, your target organ's pathophysiological functions, but also on the other hand, you have to have a very thorough knowledge of the existing modeling approaches and platform that are out there, because you do not have to reinvent the wheel. Now let's have a look at the generic organ-on-chip platform. So what we can see here is a cross-sectional view through a generic organ-on-chip that integrates a 3D tissue and a barrier tissue. And we'll start from the bottom and work our way up. So our microfluidic channels are, in this case, attached to a glass slide. Then we have a tissue compartment with a 3D tissue integrated. And of course, we need to get our cells and tissues into the chip, so we need an injection channel. This tissue compartment is separated from another compartment by a semi-permeable membrane. And now in this case here, the other compartment is perfused with medium, and it's also lined with an endothelial cell layer in order to mimic a vasculature-like channel. And then you can move on and increase complexity of your system, for example, by adding more compartmentalization structures, for example, to have another tissue compartment. There are a couple of steps that you need to consider when you start designing your organ-on-chip systems, and we think that some of the most important ones are the following. So your tissue compartment dimensions and volumes should be large enough to integrate sufficient cell numbers. So if you remember, we work in the micro scale, so we can end up with very, very little cells. But this might be beneficial if you have a very, very limited cell source, but also still you need to have enough cells to be able to read out what the cells are doing properly. Then the injection channel architectures should enable a gentle but also a reproducible cell injection. The media perfusion parameters, such as the structure of the media channel and the perfusion rates, should provide sufficient nutrition throughout the tissues. And finally, the substrate that you bond your microfluidic channels to should enable easy access for common 3D imaging techniques. All right, and now we are branching off into the two branches. For the cell sources, here it is really crucial that you identify all the cell types that form the minimal functional unit that is responsible for the targeted organ function. But you should already know that from a couple of steps before, where you did your literature research on your target organ. Also, this step is about identifying suitable cell sources for your cellular components. And there are 3 major cell sources out there, and I could actually give a whole lecture on that topic. But to be very brief, there are human cell lines, which are a scalable cell source, but they are rarely physiologically relevant because they're cancer-derived or immortalized in some other way. So these cancer-derived cell lines might be beneficial when you want to build a cancer model, but not necessarily if you want to mimic some mechanisms in healthy tissue. Then we have primary human cells or biopsies. They are patient-specific, but they can be very limited in scale and availability. And finally, we have human stem cells, which are patient-specific, scalable, and they can potentially differentiate into any cell type. However, differentiation protocols can become tricky. And with that, I am handing over to Thomas.
Okay, yeah. So now taking a look at materials, and actually I'll be doing that across the 2 different branches because there are actually some common considerations between both the physical chip materials and the biomaterials for cellular support. So the key challenge for materials that you use in an organ-on-chip development is that they have to be appropriate for your application, right? Keeping that same basic idea in mind again. And what does that mean? The chip material ideally should be biocompatible and inert. So it shouldn't interfere with biological function. It shouldn't interact with the chemicals that you put in your chip and so on. We might also want it to be gas permeable depending on how we do our culture. And importantly, we want it to be wettable because we're using microfluidics. So we're going to perfuse this with media typically. And it helps us a lot if we have wettable surfaces. On the other hand, for the biomaterial, those we want to be biomimetic and bioactive. So they provide the right biochemical cues for the cells to adhere to and to interact with, providing them an as body-like environment as possible in that regard. Tied in with that is that typically we also want them to be highly diffusive for chemical compounds and with a softness or stiffness that matches the body tissue that the cells are used to. And so most materials in our bodies, of course, being much softer than your typical chip materials in that regard. For both materials, the common consideration is that we want them to be transparent typically, because that just makes it so much easier to be able to do microscopy, which is still one of the best ways to monitor what's actually going on inside our organelle chips. Last but not least, we want the whole thing to be leak-proof. So that applies both to the chip material and actually also to the interfaces between the biomaterial and the chip material. So your liquid stays where it's supposed to. Your options there are then actually interrelated a little bit with the design that you identified that you want, right? So you identified a certain design that you want to host your cells in. Not all materials are suitable to realize all those designs. So there might be a great material out there that fulfills all your requirements on the biological side, but you can't actually make it in the right geometries from the engineering point of view. So there can always be a little bit of tension there and some trade-offs. That's important to keep in mind. But now looking at some examples, or the most common examples, just very briefly on those two sides. For chip materials, one of the classics is PDMS, or polydimethyl siloxane. It's a silicone material, and it is typically fabricated using replica molding, which is what the little sketch there at the bottom shows. It's still very popular because it's quite easy to do. It doesn't require a lot of expertise, and it tends to actually work quite well, even though there are some challenges associated also with PDMS. Another common option is using thermoplastics. So plastics like you also have everywhere in daily life. Those you have a lot of different fabrication options for, including injection molding, which is what most commercial plastics are made for. So it's actually quite easy to scale that up. And so a lot of the commercial systems that you see in the organ-on-chip market are actually made from these types of thermoplastics. But there are also lab ways to work with them. And then on the biomaterial side, again, here the idea is to be as close to, the typical biological environment as possible. And for most tissues, that tends to be a hydrogel extracellular matrix. And to mimic that, common choices are collagen, which is one of the predominant biopolymers, or Madrigel, which is this very nutrient-rich mix of biopolymers that cells tend to really like. But also you can have semi-synthetic things like Gelma that give you new fabrication opportunities while keeping some of those desirable properties. Now, the simplest thing that you can do in your system is to just apply a coating. And you can actually use these hydrogel materials. You can use also other bioproteins just to coat your chip surfaces to give your cells something to hold on to. So that's sort of the minimum approach, to just do it as a coating. But then with hydrogels, you can go into much more interesting 3D geometries also, which are the two examples that are shown here at the bottom. So 3D structures you can mold, you can template, you can laser structure. There are many different options to achieve some really cool structures with that. Or finally, one option you also have is to actually include matrix producing cells in your organ-on-chip, because biologically, that's how we get extracellular matrix, right? So it doesn't just appear out of nowhere. You actually have specialized cells that produce the matrix that then the surrounding cells actually prefer. And so doing that is a great strategy on the biomimetic side, but you do need something to start out with, right? So usually that would be a replacement strategy. Moving forward from the materials, we also, on the engineering side, take a bit of a look at sensors, because one of the advantages that we get when we use these computer chip technologies to make our cell culture systems, or even some of the simpler methods, is that it becomes actually quite easy to put sensors directly into our systems. And what that gets us is that instead of doing just single time point assays as we would typically do in traditional biological research, and that's sort of what's shown here on the far left, by having a sensor inside the system, we can then monitor in continuously, and so really get down to the minute temporal resolution over the whole course of our experiments. And so that can add a whole new dimension to the data we get out of them. For building these sensors, one important consideration is that the less stable the analyte, the closer you have to move the sensor to the cells. But then you also need to consider that some sensors can actually interfere with your cells. So again, you have these types of trade-offs. And on the right, you just see some common methodologies to integrate sensors. One of the most popular sensors right now is these electrical sensors toward the left to measure barrier integrity, which is also what the graph on the left is showing. But you can also do optical chemical sensors to get insights into what's going on with the biochemistry inside your systems. On the actuator side, right, if you can put in a sensor, you can also actually just, in many cases, run it in reverse for electrical things and actually implement actuation there. Or some very cool things can be done also with mechanical stimuli if you work with soft systems. So actually one of the quintessential organs on chips uses this mechanical stimulation for lung or gut that in your body undergoes peristaltic motion or breathing motion to achieve better biological relevance. Or for things like heart, it can be important to electrically pace it to achieve cell maturation. And so having the chip technology there allows us to implement this much more easily than in classic systems. And so with that, the pathways combine again, and it's back to Julia.
Let's inject some cells and tissues. And surprisingly, there are a myriad of things that you have to consider if you want to inject your cells or tissues into the microfluidic platform you just developed. Among these considerations are what cellular composition, is there a tissue stratification, what is my tissue geometry? Then you need to also consider physiological cell density, cell-to-cell ratios, and cell-to-ECM ratios. And that usually leads to a very customized injection process for all the individual organ-on-chip systems out there. So you'll probably have to dedicate a lot of efforts on how do you get your cells into your chip. However, there are 2-3 general strategies of how you can get your cells or tissues into the chip. Again, we start off with the human body, and if you remember from the beginning, it is a cell source for the cells that we integrate. We can either get biopsies from the human body or stem cells directly. If we then isolate single cells from the biopsies or differentiate single cells and different cell types from the stem cells and puzzle those back at the ratios that we want into our organ-on-chip system, we have a bottom-up approach. We can also use a building block assembly. That means that we first make building blocks such as spheroids or organoids from our single cells and integrate those into the organ-on-chip system. And as a third strategy, there's what's called an explant integration. That means we chop up our biopsy into microfragments and integrate these into our organ-on-chip system. And of course, you can also combine the different strategies to set up your organ-on-chip model. Further things you have to consider is whether you have a compartmentalization on your chip. Then you have to ask yourself, if I have different compartments and I have different cell types, which cells do I inject first and which cells come after? So you see, there are many, many things that you need to consider when you want to integrate your tissues into the chip. But then when you finally have your tissues in the chip, you want to know what's happening to your cells. So you need to establish a toolbox of different readout methods that allow you to investigate generic readouts on the one hand. So, for example, the viability of your cells on the chip, as well as their morphology. But you also want to have a look at organ and application-specific functional readouts. For example, barrier integrity or endocrine activity or contractile forces, for example. So there's also really a lot of work to do to establish readout methods that allow you to see what's happening to my cells on the chip. If you have done that step, the last step is the functional validation of the model. And here you really need to very comprehensively characterize your model's capability to recreate the in vivo functions and mechanisms you aimed for in the beginning of the process. But you also need to know the limitations of your model in order to define the window of functional stability. Within that step, you will probably run many studies that depend very much on the specific intended use of your model. And the overall aim of that step is to build confidence in the predictivity and translatability of the results that you obtain with your model. And then finally, your model will be able, or your model is ready to be applied, for example, in mechanistic biomedical research, clinical research, or pharma R&D. As a last step, a couple of take-home messages and some advice from Thomas's and my side, also including things that we wish we knew before we started to work with organ-on-chips. Context of use really is key. It can be really tricky to develop a model without having a context of use defined before you start working with it, because you might have a fancy model, but then it's not useful for anything. And you wouldn't want that, I guess. Also, organ-on-chip research is really a highly interdisciplinary effort. So what I learned in the past decade is that it really helps to build very strong collaborations, because you cannot be an expert in all of the different disciplines that your organ-on-chip project will span. And it's also really worth it to invest time and efforts to establish very clear communications with your collaborators, so that you can really be sure that the different partners in the project understand one another, and then you get the maximum output from your project.
And then on the sort of lessons learned side, one of the things that you get in organs-on-chips is that you're combining highly advanced biology and highly advanced engineering, which both already have many different things that can go wrong with your stem cell culture, with your fabrication process. But then when you actually put them together in an organ-on-chip, you discover all these new and exciting things that can start to go wrong sort of emergently by now having to put these things together. And so that type of troubleshooting is, what really eats up a lot of time in this field. Some of the maybe less obvious pitfalls there are that, when you try to build these complex co-cultures of different cell types interacting, it's always important to keep in mind that the cell types might have complex and even competing requirements in terms of the cell media or the ECM that they require. And so that can actually be very difficult to put all the cells that you want to have interacting on the same chip. In your bodies, that's nicely regulated, but on a chip, we just can't approach that type of interaction quite yet. Another thing is the chip-to-world and also chip-to-chip interfacing. So if you want to have multi-chip systems, how to put them together without introducing huge dead volumes between the chips with tubing and so on. And also your chip very rarely just is a simple chip, right? You will have some type of pump system also around it that you also need to consider. And especially when you work with tube systems, it's actually very important to also consider the materials of those tubings, how exactly the pump will interact with your cells and so on. So don't just think about your chip, think about the whole system that you're really building. Maintaining sterility for these organs-on-chips also tends to be more challenging than your classic cell culture, right? Because now you have all these different parts and all these inlets and outlets. You don't just have something that you can nicely close off typically. And so that can be very challenging, and especially if you do your own biomaterial development along the side, that opens a whole new field of potential issues there. And everyone's favorite challenge with organs on chip, of course, over the years is air bubbles and air bubbles everywhere. That is just what really disrupts a lot of organs on chips and one of the big challenges. But despite of it, I think we can still say that we're having fun in the field, and it is a really cool area to be working in. And with that, thank you for coming and thank you for attending, and we look forward to your questions.
Before we jump into the question, I think we have one last...
Oh, that's right. We did have one last poll prepared. Sorry about that.
Yes, so we would really like to know from you now, after listening to our webinar, which are the biggest hurdles for you to overcome when starting to work with an organ-on-chip system? We see the answers, and they are quite spread about across all of our answering options. I was actually thinking maybe we should have also added a nothing option, that now after our webinar, maybe everything is so clear, but apparently not. Okay, so we have 30% have as the biggest hurdle to define the context of use, 25% coming up with a concept and or design, 32% the microfluidics part. Identifying and preparing cells doesn't seem to be that difficult because I only got a 13% vote, but then culturing the cells on the chip and reading them out got 35% and 40% respectively. And about 20% are just overwhelmed by everything with organ-on-chip.
All right, and we already have a lot of questions, so I would say we don't waste any time and jump directly in it. And I'm really, really happy about especially also the first question, and I will read it out to you. So where can one actually learn how to produce these chips?
Yeah, where can you learn how to make them? Excellent question. It's as Julia said, it's a very interdisciplinary field, and so one of the best options perhaps is to see if there are any people working on microfluidics at your institution, and then they can likely give you some input on the different fabrication techniques for these types of systems, because from the engineering point of view, it is more or less a specialized microfluidics problem, right? And so a lot of the fabrication techniques are going to be very similar there. If there's no one that you can find to work with, one option that I think we also have a question on later is that there are actually commercial systems. So if the commercial system fits your context of use, that can be a simple option to sort of circumvent this. Another good opportunity is if you can manage the European Organ-on-Chip Society actually hosts a summer school, and so where we cover these types of topics. And so we would be happy to see you attend there next year.
All right, then we move on to the next question, which also deals with the material. So the question is, what are the challenges using PDMS as the chip materials, and what are the workarounds?
So I can also take that one very briefly. So it strongly depends again on what exactly do you want to do with your chip. So for some applications, PDMS is perfectly fine. So the issues and benefits of PDMS in a way is that it's a very loose polymer matrix. So that means on the one hand, it's permeable to gas. So that can be beneficial for CO2 control from an incubator environment and to ensure sufficient oxygen is present and so on. For some cells, you might not want that. So again, it depends on what your context is. So, but the loose structure also means that actually it is prone to soak up small molecules to some extent. And that, again, depends. Do you want to study a small molecule drug at very low concentrations? Then maybe PDMS isn't for you. But if it's a large molecular protein that you're studying as your compound, it might be less of an issue because proteins cannot go directly into the PDMS matrix. It also comes down to the surface area to volume ratios, because if you actually have a continuously perfused system where you continuously deliver fresh drug, then also the PDMS absorption isn't that much of an issue. And to alleviate that, there is a wide range of literature that deals with how to deal with PDMS absorption, how to deal with PDMS bonding, that's another thing. When you want to stick PDMS together with some metals or plastics, that can be a challenge, and other materials are better. But again, there are protocols to do it with PDMS. Everything comes down to what exactly do you want to do with it.
Since you also mentioned the commercial organ-on-chip systems, there is a question related to that. It reads how to decide which commercial organ-on-chip system to choose if budget is limited.
I would say that really depends on your context of use. There's quite a variety of commercial systems out there, but many of them have different designs. So they are advantages for some intended applications, but not necessarily super advantages for other applications. So I think as a first step, you have to become clear of what will be, or what do you need your model for? Do you have, for example, a barrier system? Then probably you would want to go with a chip that provides you with a barrier already that you need to seed the cells onto to create your cellular barrier, but you'd probably want to go for a model that has some membrane and then two compartments, like, for example, the Emulate chip or the Dynamic 42 chip. If you want to study vascularization, for example, then I would rather go for other commercially available chip systems that are out there, such as the Biotech chip or the Mimetas chip, for example. So I would say it really depends on what you need your model for and what you want to recapitulate in vitro.
I have a question. So there is somehow some good database that people could use to look indeed of this characteristic, what is available, or based on the context of use, what is already was applied. Because I can imagine it's much information. There is any hub when one can find this information? Not all, but some.
I think I forget what it's called, but the US program for organ-on-chip validation, they started a database where you can register your organ-on-chip experiments. And so that they also include, I think, some commercial systems in that. So that might, have some of this information, but it's been a long time since I looked at that, and I'm not sure how open it is and how suitable it is to actually answer that question. So building something like that would certainly be very helpful to the field.
All right, then we have two questions which are more on the biological side, so probably more towards Julia. So the first one is, do you need a written consent of the cell donor? Does the donor have any rights for royalties on the results gained with its cells?
Okay, so first question, yes, definitely. So whenever you want to work with cells derived from human beings, you need to get their consent. You need to get an ethics approval through your university, probably, that you're allowed to work with these cells. It is very, very tightly regulated. You also, of course, do not get a lot of information of the donor, which can sometimes become a problem. But of course, it needs to be anonymous. We as researchers should not be able to trace back who was the donor of these cells. And the question about the royalties, since everything is anonymous, I don't think that the donors of the cells will get any benefits from donating, other than helping progress in research and helping us researchers to perform our experiments.
All right, thank you, Julia. Then maybe the second question I mentioned. So how many donors do you need to cover the whole human population, races, ages, sexes, etc., to predict effectivity to avoid pitfalls in clinical studies?
So that is a very tough question. I think the larger the sample size that you have, the more different aspects of the population you'll be able to reflect, the better. But that is really a question, I think, that some toxicologists might be able to answer a little bit better. Because, I simply can't say. Probably also depends on the specific scientific question that you have. What do you want to study? What I can say is that mostly organ on chip is not very high throughput, especially if you get into a very complex model milieu where you have many different cell types, where it takes you a very, very long time to prepare the cells, to inject everything, to keep everything alive. So, and then you also want biological replicates for each individual donor, so that you have a representative sample size that you can say that the results that you obtained with your model are really, really reliable and robust. So it can get really tricky to run several donors, because also most of the time you want to run the experiment at the same time to have all the same culture conditions and so on. So in my experience, organ on chip is not a super high throughput technology.
All right, thank you, Julia. Then maybe a little more towards Thomas' side. Between the optical versus electrical, for example, extracellular potential monitoring, which one would be more suitable to analyze the biological function of the cells inside the chip, and in which context? If they both might be used together simultaneously, what are the concerns?
In keeping with a continuing theme, which one you choose will depend on the context of what you actually want to assay in your system. I mean, broadly speaking, the purely electrical ones are going to be most useful when you're working with electroactive cells. So when you have neurons, when you have cardiac cells, then purely electrical measurements give you a lot of information about the cells. For barriers, purely electrical measurements are also quite interesting because they can give you a very simple measurement of the barrier status. Optical measurements are interesting when you want to look at morphology, right? So for analyzing morphological structures, or for things that require immunofluorescent staining and tracking, that cannot be beat with other methods, because you get both spatial and temporal resolution if you do optical. But more broadly speaking, right, if whether you want to measure glucose using an optical sensor or an electrochemical sensor will mostly depend on, what your chip architecture is, where exactly do you need to measure, what are the diffusion distances, can you get wires into your chip, that type of thing. So some sensors you can go either way, some there's an obvious candidate. It comes down to that. Using both simultaneously, the thing to watch out for is that electrical and electrochemical things tend to, use non-transparent electrodes, gold, things like that. And so that would obviously interfere with imaging, but there's also ways to get around that.
Next question, are nanofibers used in production? Can you tell us about its uses in the field of neuroscience? Maybe a question for the both of you.
So I can start at least. So there are applications for nanofibrous biomaterials, certainly, and especially in neural culture and neural templating also, that can be a beneficial characteristic of your biomaterials. So one common culture material for, or one that tends to work quite well for neural cultures, it turns out, is actually a cellulose fiber material. So things like that certainly exist and are certainly in use, especially when it comes to neural applications, because they tend to like these fiber structures.
Then we have a question. Oh, I like this one. How do you stay up to date with new fabrication methods for organ on chip? Any journals that are a must subscribe?
So maybe I can answer general, and then Thomas adds to that from the fabrication the perspective. So I usually get or stay up to date by attending conferences, mostly because this usually gives a very, very broad overview. Also, since Organ on Chip is very interdisciplinary, it also helps to get an overview. It helps you to dive into maybe other organ models that are not necessarily the organ that you're working with that you would look for if you look online. So from my experience, I get most exposure to, in general, new methods in the organ on chip technology, and also new fabrication methods by attending conferences, online seminars or webinars, and so on. I think soon there will be a special organ on chip journal through the MPS World Summit, but maybe Thomas.
Yeah, so to just briefly follow on that, I agree that conferences are really one of the best ways to go, because you get a nice, great breadth, and you get, you know, the newest research, and ideally even things that haven't actually been published. So yeah, come to EUROX and also the MPS World Summit. Other than that, as Julia said, it's so interdisciplinary, so there's no single journal that things end up in, right? I find that Google Scholar alerts are a great way to go for things that you have an interest in. For specifically fabrication things, I mean, there are for sure some journals that, you know, have more of that, your typical suspects for when it comes to engineering type things, microfabrication and nanoengineering, or advanced materials for maybe also more biomaterial things, advanced healthcare materials. Yeah, but honestly, the list of journals that good organ on chip works ends up in is so broad that, yeah, it doesn't really make sense to just follow a single journal. Yeah.
All right, thanks you two. Then we have two more questions. Do you have any comments on the physical environment of the organoids? Means, what do you think about physiological oxygen tension and mechanical stimulation? Shouldn't we try to be more physiological in these terms, in addition to material structure and organization?
Yes, definitely. I agree that this maybe we haven't covered too much in our webinar today due to time. That is also a lot about manipulating gas permeability in general to the chip, as well as mechanical stimulation. Thomas touched upon this when he was talking about actuators. It really, again, depends on what types of organoids you have, or what types of tissues you have in general on the chip. I know that there are liver on chip systems out there that really look into or managed to establish different zones of oxygen concentration to really be more representative of the in vivo environment of the liver. But yeah, that really depends. For organoids specifically, I guess it depends on also what type of organoids you're working with. So I don't know, maybe Thomas, do you have?
I don't have much to add to this. I mean, for organoids, maybe this question is also trying to get at the vascularization issue for organoids, which is, of course, a big, you know, bioengineering challenge. You can approach it both bottom up, top down, many ways to go about achieving more physiological oxygen distribution in your tissues. But that in itself would cover a whole other webinar, I think.
Yeah.
All right, thanks you two. Then last question, organ on chip sample, specific standardized protocols are established, or are they still under research process?
So I think that question is going the direction of if everything is so customized for each individual chip, how can we still make sure, or can we make efforts to make it more standardizable so that it will be easier to transfer one chip that was developed in, or one system that was developed in one lab to another lab? I know that there are a lot of efforts being made to have things a little bit more standardized. For example, when it comes to chip-to-world connection, where are the ports located on my chip that make it, for example, easier to integrate it with an external pumping system, or where are my channels located in the microfluidic platform? Can that be standardized to make imaging easier, for example? Do you have any examples for standardization?
I mean, the commercial players, right? They will have some good protocols for their chips. So Emulate will give you a very nice procedure to follow. Mimetas will give you a very nice procedure to follow. But again, it's, you know, for a specific context of use. And if you use your biological system with the existing commercial chips, you might still have to adapt it. And yeah, it is for our field, I think, really critical that the protocols are always documented very clearly in the papers, so that people can actually follow and reproduce those steps.
All right, that was a really nice discussion. Unfortunately, we are running out of time. I wish we could just continue, but we can't. So thank you so much for this great talk, and also for the discussion. And we would be super happy to welcome you again for the next webinars. As always, registration is free, and you can just do it via our website. And we are looking forward to welcome you next time. And for today, we wish you a nice evening, and thanks for attending, and hopefully see you soon. Bye, everyone.
Thank you. Bye-bye.
Bye-bye.